Welcome to Biology Made Easy. In this lesson, we are discussing water potential values as we find them in transports of water. Welcome. Well, first of all, what is water potential? And we want to say is the tendency of water molecules to move from one place to another. And the tendency is such that water moves from an area where it is more concentrated to areas where it is less concentrated. So where it's more concentrated, has high water potential. So it moves from regions of high water potential to regions of low water potential. Now, the movement of water molecules is not necessarily from one cell to another. We can have water moving from a cell into the external environment. As you can have in the leaf, you have water molecules as water vapor moving from the intercellular spaces into the outside air. So we have high water potential in the intercellular spaces and low water potential in the outside air. Now, not only that, we can have water molecules moving from an external source like the soil into the roots. And we can then say that there's high water potential in the soil water and low water potential in the roots. As we can see, you have soil water moving into the root hair by osmosis, because there's more water molecules here than here. So higher water potential in the soil water than inside the root hair. Well, not only that, we can have water moving inside the cell from one point to another. You can have a tube, you can have a vessel where, like the xylem vessel, where this region A will have more water molecules than compared to this point. And so water molecules move from point A to point B, as you can find in xylem vessels. This is a xylem vessel, and we know water molecules move from below to up the xylem vessels. And so the question is, what creates the high and the low water potential? What is it that makes water potential low and high? And it's the ratio of solutes and water. Well, you have these two cells. We want to use it to explain. The solute is the yellow, when water molecules are the red bits. Now, here in this cell, you have less solutes and more water. You have more solutes and less water. So the ratio between amounts of solutes and water molecules determine the water potential. So here you have more water. In this system, you have more water and less solute. So you have high water potential in this system. And here you have less water, more solute. So you have low water potential, less water potential. So the direction of movement of water from the high water potential to the low water potential areas. So that is it. Now, having looked at it like this, we want to find out how water potential is measured. Water potential, you know, is a force. Water can only move when it has a force to move. And so the force, Pascal, is the units. And 1 Pascal is equivalent to 1 Newton. And pure water is set as the base of the measurements, because pure water can move into any system. You have pure water here, and pure water has the tendency to move into any solution. So pure water, by convention, is said to have the highest water potential. And the highest water potential, pure water, is set to be 0. And so every other solution will be less negative, because every other solution will have lower water potential. And if the highest water potential is 0, then all other solutions will have a negative value. So when you have a less concentrated solution, it means it has plenty water. And so you have a high water potential. So you have less negative, because then the value is more closer to pure water. Then you have more concentrated solution. It means you have little water. So you have low water potential, and the value is more negative. We've put down hypothetical values as this. Now you have a highly concentrated solution. It means it has very little amount of water molecules. So we have low water potential, and the value is greatly negative, like as we have put as this. So we go back to look at our solution, and here you have less solutes, you have more water, so you have high water potential, and we can give it around minus 200 pascals. This is low water potential, so it has lower negative, so it becomes minus 500 pascals. This is how water potential is measured. Now we want to look at the water potential values in the plant during transport of water. So we have drawn here a section of a leaf and put, this is the veinlet, this is the petiole, which is also a xylem, and we have xylem of the stem, xylem of the root, and you go and get into the root hair and the soil water. Now we have put these lettering A to K as points to show points on the plant. For instance, point A, we have said point A is the air outside the leaf. Point B is the intercellular spaces here. Point C is the tip of the vein. And the stem, we start with D and E are all points on the plant. And we come and look at the soil, which we have given it point K. We are going to look at water potential values that concerns the water potential gradient down which water is pulled up the plant. The water potential values have been brought here to make it easier for us. Outside the leaf here has the lowest water potential, because the water evaporates from the intercellular spaces and moves out. It means the water potential in the intercellular spaces is higher than the water potential in the air around the leaf. So around the leaf here has the lowest water potential. Water is going to move from the intercellular spaces, and water is moving from the vein where the water potential is higher to fill the place where the water has evaporated. So we know that the water potential value is the least in the outside, is greater in the intercellular spaces, but is greater in the vein by which the water moves. So we know that where the water potential is the least will have the lowest water potential, and it will be the greatest negative because it's much, much further away from 0. So in the plant, the lowest water potential value is the greatest negative. So we have here, look here, the hypothetical figures we have given. Water potential value outside the leaf here is the lowest, and it will have the greatest negative, which we have hypothetically given 15,000 pascals. And the water potential value in the cellular spaces here will be higher than outside here, and we give it minus 10,000 pascals. And as water moves on from a region of higher potential in the vein into the interstellar spaces. So in the tip of the vein, water potential value will be higher, minus 9,000 pascals. And that is how the water potential values become higher. And as it becomes higher, the negativeness of it decreases. So you come and get to the soil water that has the greatest water potential value, meaning it's closer to zero. So you see, it's the least pascal. So this is how the water potential gradient is formed, and this is how it is described as water is pulled down a water potential gradient. You see, very neat. From low water potential to high water potential. But then, as far as Pascal's values are concerned, it's down. So, if we've made things clearer with this explanation of water potential values, look ahead in our next lesson. We are also going to explain hydrostatic pressure values and tension values as water is transported from the soil water into the xylem vessels up through the whole plant. So till we meet in our next lesson, if you found the lesson good, please give us a thumb and please subscribe. Thank you very much. Goodbye.